Yes, and let me just hide you guys. Okay, cool. So I'll be talking about understanding E. coli survival during composting. So this project stems from Dow's work that investigates microbial risks associated with amending soils with manure and compost. And also this project was done in collaboration with Dr. Amy Kane and her PhD student, Belinda Fabian at Macquarie University. Organic amendments, such as animal manures, are a rich source of nutrients and is commonly used to amend soil. There are many purposes for composting manures, including degrading chemicals that may have been introduced into manures, killing pest, weeds, and insect eggs, and increasing availability of nutrients. Of interest to us is reducing the load of microbial pathogens so that we can use compost to safely grow fresh produce. In our previous student sessions where Dao was presenting her work, she found that E. coli can survive composting conditions by becoming viable but non-culturable cells, called VBNCs, which is a state of hibernation that cells can enter as a way to survive stress. Dao also found that under the right stimuli, these VBNCs can also exit dormancy and then resume growth. So the question that we wanted to answer here was, how does bacteria survive composting? Because if we can understand this, we can use this information to improve the composting process to reduce pathogen load and prevent the growth. And to do this, we use a method called transposon sequencing to find the genes and cellular processes that are required for bacteria to survive composting temperatures. To give you a background about transposon sequencing, this technique uses a library of mutant bacteria where each bacteria cell has a gene that has been disrupted in function. The library is subjected to different environments and fitness of mutants are tested. Mutants that are able to grow have genes that are beneficial to survive and therefore they have a higher fitness. And the cells that cannot grow don't have the genes that are required for survival and therefore they're essential for growing in that particular condition. So in terms of the methodology, we got our mutant library from our collaborator Amy, and we grew it up and incubated them with and without the presence of nutrients at composting temperatures according to Australian guidelines, which stipulates that it's growth at treatment at 55 degrees for 3 consecutive days. We then extracted the DNA from the mutants and then sent it off to Ramashore Center for Sequencing and then analyzed the data that came back with the help of Belinda. To answer the question of how bacteria survives in composting, we took a look at 2 different conditions. The first condition investigated survival in heat, namely what genes are essential and also beneficial to survive temperatures of composting only. And so this condition will help us understand bacterial survival in the second condition, which is our laboratory mimic of the composting process, which is slightly more complex with the presence of both heat and also nutrients. And so for the rest of this talk, I'll only be focusing on how bacteria survives in heat. So in the first condition, we have 78 genes that contributed to survival. And then when we put these genes into an interaction map, we see that the genes cluster and then these clusters connect together, which suggests that they're involved in same functions that affect or are needed by other processes. If we take a look at the description of the genes on this map, we see that the essential genes are related to RNA, DNA, amino acid synthesis, the generation and use of energy, and also processes related to the cell surface as well, which you can see, which has been highlighted with the arrows. We also see transcription regulation, which is similar to a master controller that turns on or off cellular processes in both essential and also beneficial genes. So what does this all mean and how are these functions important in heat survival? Well, DNA and proteins are needed for cells to survive. If there's no DNA and if there's no amino acid, proteins cannot be made, and proteins are the workhorses that drive cellular functions. Making DNA and also amino acids require energy, which needs to be generated, and one of the ways through how this is done is through an enzyme complex on the membrane at the cell surface. Processes related to the cell surface, such as those to do with the membrane and also the cell wall, are essential for survival as it keeps the cells intact. So breaches in integrity of these structures will cause cell death. And as such, the cell surface has a critical function in sensing changes in the external cellular environment, such as the heat and osmotic changes, which then enlists the help of transcription regulators to enable cellular processes that ensure the integrity of the cell surface. So now if we switch into the beneficial side, where we take a look at the regulators that are beneficial to survive heat, what we also see is that cell surface related processes are also present as well. And this is quite interesting as the genes that are beneficial and also have and give a fitness advantage in surviving heat are most likely the ones that drive dormancy. While we don't see these essential processes such as energy generation and also DNA and amino acid synthesis in the genes which will benefit heat survival, these processes have been reported in heat-treated bacterial cells and also in PBNCs. So what this may suggest is that cellular processes that are critical for heat survival and cause cell death if you target these processes may also drive dormancy as an alternative survival strategy. But of course, we do need more molecular evidence to know if this occurs and by which means this occurs. So to conclude, the maintenance and the integrity of the cell surface, as well as energy generation and use, and also the making of RNA, DNA, and amino acids are critical for E. coli to survive in heat. We also see that these processes are quite connected, which suggests that they require one another for function. What we see in the literature and also in our data, it suggests that similar processes that are critical for surviving heat also play a role in inducing PBNCs. And we need to understand where the similarities and also the differences are if we are to improve the composting process. So finally, to answer the question of how E. coli survives in compost, we still need to look at condition number 2, essentially into the genes that are required for survival in both the presence of heat and also nutrients, which is a simple mimic of composting. And then also do the real thing, perform transposon sequencing in actual compost, where conditions are much more complex. And that's the end of my talk.